What's up guys, welcome back to my YouTube channel, and in today's video I'm going to tell you exactly what do you need to know about oxidative phosphorylation so you can answer any question about this topic. Just remembering you all that this is a video that is part of a playlist where we talked about bioenergetics. So we have already covered glycogenesis, link reaction, Krebs cycle, and finally we're going to finish with this beautiful thing. And if you enjoyed this content and appreciate what I do, please just take 2 seconds to smash that like button. You have no idea how much this helps the channel grow. And if you're new to this channel, make sure to subscribe because we're going to have a lot of good content in the following days. Alright, so let's not waste any more time and let's dive into it. Alright, so first thing first, let's talk about where does this take place? Where does it happen? So we're talking about the oxidative phosphorylation. That is one of the most important processes for productions of ATP. So the big guns are going to be produced through this process, okay? And it's going to happen in the mitochondria. This is a very important organelle that are present only on eukaryote cells. And this organelle is going to have a very particular shape because it's not only going to have one membrane, but it's going to have 2 membranes. As you can see, there's an outer membrane and an inner membrane. And the inner membrane, it's kind of like folding into itself, okay? We're going to call this the cristae. But these are basically membrane, just like the other ones, built up by phospholipids. Now, just because I like to bring things into context, there is a particular reason why we have all these entrances in the inner membrane, and the reason is so we can the proteins that are going to be important on the electron transportation. I'm going to show you one example so we can explore a little bit what happened. But imagine that along this inner membrane, there's so many of these group of proteins to maximize the productions of ATP. So yeah, just for you guys to remember that glycolysis is the only process of the cellular respiration that is going to happen outside of the mitochondria in the cytoplasm. All the other ones going to take place around here. So we're going to have the link reaction, for example. That one is going to take place entirely at the core of the mitochondria that we're going to call the matrix. The Krebs cycle is also going to happen here, but there will be one important enzyme that's going to be attached in the inner side of this membrane. But in general line, it happens at the matrix of the mitochondria. And finally, we're going to have the oxidative phosphorylation that is going to happen with the help of the electron transporters located in the inner membrane of the mitochondria. Alright, so this all might seem too simple, too obvious for you all, but don't forget that the IMAT is an exam that can be really straightforward with the questions sometimes. So make sure to know exactly where does each one of these processes happen at the mitochondria or the cytoplasm, and what are the mechanisms involved. Alright, so let's imagine for a second that we extract a small segment of 2 of the mitochondrial membranes. Okay, so we're going to have the outer membrane that is going to be in contact with the cytoplasm, and the inner membrane in contact with the matrix of the mitochondria. And in between, you're going to have this very important space that is the intermembrane space. And this is going to be a very important process, as I mentioned, but now saying more technically, because it's going to convert the potential energy stored in the electron carriers, so the cofactor enzymes, the NADH, the FADH2, remember them, into a chemical energy of ATP. So it's basically saying that we're going to produce chemical energy through the energy that are stored in the electrons, more specifically on the NADH and the FADH2. If it seems too complicated and confusing, don't worry, because there's a reason for that, because this is basically pure chemistry and physics combined with biology. I'll tell you what, your first year of medicine, you're gonna have a lot of fun. But alright guys, seriously, there's no reason to panic, okay? I want to show you this. This is basically what you need to know. This is an image of the electron transport chain, and this is basically it, okay? I'm not going to dive into every single detail because for the IMAT, there's only a couple of things that you need to know for you to be okay. So these are the things I want to focus in, but of course, I need to bring some context as what's going on here. So what are we seeing? As I mentioned, in the inner membrane of the mitochondria, we're going to be able to see some enzymes and coenzymes all working in order for us to be able to produce ATP. How does that happen? Alright, so you already know that the main characters for this to happen are going to be the NADH and the FADH2. Where they're coming from, basically, the NADH, we can have a bunch of them from the Krebs cycle, remembering that for 1 molecule of pyruvate, we can produce 3 molecules of NADH. Just multiplying for glucose, we can produce 6 NADH in the Krebs cycle. Don't forget that we also produce 2 NADH in the glycolysis. Remember, glycolysis is easy, 2, 2, 2. We produce 2 NADH, 2 ATP, and 2 molecules of pyruvate. And the FADH2 is basically coming from the Krebs cycle, remembering that we produce 1 per pyruvate, 2 per glucose. That's the main source of it. Okay, so the first important thing you cannot forget for the IMAT, okay, this is mandatory, is that the NADH, whenever it's produced, okay, NADH, remember, what is the H for here? Is basically saying that this molecule is reduced, is carrying an electron. The NADH will always come here and bind to this enzyme here in green that we're going to call complex 1. And what's going to happen? It's going to drop the electrons on this complex 1, and once it drops the electrons, you know what happens? The NADH becomes the NAD+. That is what it is oxidized. It's an oxidized version. So it was reduced, carrying the electrons. Whenever it drops it, it is oxidized, okay? I'm just framing this little part here because oxidation and reduction is something that IMAT loves to talk about. Alright, so after that, we're going to have a very very similar process, but instead of NADH, we're gonna have the FADH2 dropping their electrons to this complex 2, this enzyme in orange, as you can see here. Once that's done, we're gonna have the formation of FAD, that is the oxidized version of the FADH2, and whenever we have FAD and NAD+, okay, these oxidized coenzymes, they can go straight back to the Krebs cycle to restart another cycle again. Now, what happens next is quite interesting if you think of a hot potato thing, you know, because complex 1 has electrons and complex 2 as well, but neither of them want to stay with the electrons because they're going to feel a lot unstable, you know, and it's not natural for them. So they're going to pass on to another coenzyme. In this case, the coenzyme Q, also known as Q10. You know, some people take this as a supplement, but it's going to be important as one of the electron transporters. You can also find a different name for this coenzyme that is ubiquinone, but the most important thing is that whenever it has the electrons, it's going to pass on to complex 3. But before we move on, because basically is that, what are the things that you need to remember? Like, you can never forget that the NADH will always drop their electrons on complex 1 and the FADH2 always on complex 2, never the other way around. You know, there's so many questions, and because there's not much to ask about oxidative phosphorylation, the questions are almost all the same. You know, they try to trick you and make you mix up the order of each one of these cofactors. So saying that FADH2 will drop their electrons at complex 3 or complex 1, but no, it's always going to be complex 2. So this is the first thing you need to keep in mind. Other than that, something that I haven't mentioned yet, but that crosses a bigger difference between complex 1 and complex 2, is that whenever there are electrons being dropped on complex 1, what's going to happen simultaneously is that we're going to have some hydrogens being pumped out to this intermembrane space. That's not going to happen in complex 2. You can even see here that the complex 2 is a little bit shorter, you know, it doesn't make it to the other way out. So it's only going to happen in complex 1 and the other complex that we're going to see in a minute. But alright, for the next step here, there's no really mystery, okay? Whenever we have the electrons present in the coenzyme Q, it's going to be transferred to complex 3. Whenever the complex 3 has these electrons, it's also going to be a little bit uncomfortable with it, so it will want to pass on. But before doing that, complex 3 will be pumping protons to the intermembrane space, and the one responsible for capturing the electrons after complex 3, it's going to be the coenzyme cytochrome C. Now, one thing interesting to notice here is that cytochrome C and coenzyme Q, they're both coenzyme, just like the other ones, but the difference is that they're capable of moving. So they're actually being a bridge and connecting the electron transporting along all these coenzymes. So okay, we have the cytochrome C carrying these electrons. The next thing that's going to happen is that it's going to be transported to complex 4. Complex 4 will be the last coenzyme to have to deal with these electrons because, I don't know, he gets fed up with it. The next thing that's going to happen is that finally, when the electrons arrived here to complex 4 after being passed on and on, it will finally find its final acceptor, and this little guy here won't let him down. It will keep him for a lifetime. Alright, so just to recap here, what happened? We had the NADH dropping their electrons on complex 1 specifically. Complex 1 will pump hydrogens to the intermembrane space, and after that, the electrons will be transferred to the coenzyme Q. The FADH2 specifically will only park in drop their electrons on complex 2, okay? There will be no hydrogens being pumped to the intermembrane space. This is the only coenzyme that does not contribute for the gradient of hydrogens, but it will also drop their electrons to coenzyme Q. So coenzyme Q is like this bridge that's going to communicate these two complexes, okay? And passing on to complex 3. Complex 3 and complex 4 will be responsible for pumping hydrogens to the intermembrane space, but complex 4 is going to be especially important because he's going to be the one that's going to put an end to this hot potato story. All right, now this might be one of the most crucial parts because so far we have not created any ATP. We're only talking about transporting electrons and accumulating a bunch of hydrogens, protons in this intermembrane space. Now, the real work is going to happen with the help of the ATP synthetase. This process we often call chemiosmosis, that is basically this accumulation of protons creating this gradient, chemical gradient, okay, that we often call proton motive force. Proton because it's basically an hydrogen without an electron, so that's basically a proton, and a motive force is because it's going to be actually the motor that will produce the ATP at the very end. So you guys know that there's a lot more hydrogens in the intermembrane space compared to the matrix of the mitochondria. So the natural flow here would be for the hydrogen to escape, okay, get rid of this space, or go to the matrix of the mitochondria, or just go to the cytoplasm. But there's no way out, and this happens all along the mitochondria, okay? The only way out is through these ATP synthetase. So that's basically what's going to happen. The hydrogens go straight through the ATP synthetase following its gradient, almost like water in a dam. And the flow of these bunch of hydrogens going through the ATP synthetase, the only place they can go, will actually create a spin in this enzyme that will be strong enough to promote a very endergonic energy reaction. Now, I'm gonna be honest with you guys, this part here is very interesting, but also very detailed and complex, you know, but you can leave that to the medical school. It's true that there's so many bits here responsible for doing different things, but the way I want you to look at it is just like a motor. Flow of hydrogens through the ATP synthetase will promote this spin that will actually create this endergonic energy. And when I say endergonic, it's important for you to remember this word. Okay, this is one of the most important words, because if it says something like the ATP synthetase happens through a reaction that release heat, that's completely wrong, because that is the description of an isorganic reaction. The endergonic, you're going to have to absorb energy for this to happen. So yeah, that's it. That's going to be the end of the story. You're going to have these complex 1, 2, 3, and 4 that are part of the electron transport chain. We have the specific names here, but they're not important to remember. When we're talking about the ATP synthetase, this is actually the guy who is going to produce a lot of ATP, and usually it's associated with the chemiosmosis process. That is that concentrations of hydrogens all accumulated in the intermembrane space. Now, it is important for you to know more or less how many ATP we can produce through all this process of cell respiration. We're going to talk about this in a second, but I told you that the hydrogen cannot escape, and that can only move in to the matrix of the mitochondria through the ATP synthetase. But that's not completely true, okay? Because we're gonna have some molecules that are called Thermogenin, that it's basically a natural molecule that we can find a lot in babies, also in some bears and other mammals. I know this really looks like an advertisement, like I'm trying to sell you Thermogenin. But the thing is, we're going to have in some of our cells some special types of mitochondria carrying these Thermogenin proteins. What they do, they're actually like a leak in the membrane of the mitochondria. So the hydrogen can actually escape through another spot, okay? Instead of escaping through the ATP synthetase, it can go straight through this pore that we call Thermogenin. Why does that happen? If you think about an energetic point of view, it is not really efficient, because as more hydrogens that we have available to go through this ATP synthetase, more ATP will be produced. But if you think about on babies, especially other animals that has to find a way to produce heat, this is the solution, because whenever you have hydrogen going through this protein, there will be a lot of heat being produced. So this is a natural way that our body found to produce heat whenever it's necessary, you know, beside all the other ways we can produce heat. The cells that usually carry these special mitochondria are called the brown fat cells. This is the main example, and newborns usually they have a bunch of it on their back, on their chest, and so on, and other mammals also has a bunch of them as well. All right, so for us to have an idea of how many ATPs we can produce with cell respiration, we have to go back to glycolysis and start counting all those NADHs and FADH2 we produced along the way. Okay, so we talked about this, so you guys already know, but the important detail here is that roughly we're going to be producing 2.5 ATP per molecule of NADH and 1.5 ATP per molecule of FADH2. So you can see that NADH actually gives you a lot more ATP compared to FADH. Good thing is that we're only going to have a production of 2 FADH2 in Krebs cycle. You should also not forget that we have some ATP being produced on a substrate level. Okay, you're not talking about oxidative phosphorylation, you're talking about a substrate phosphorylation. This is important to distinguish that maybe the IMAT might talk about. Glycolysis, we produce 2. And Krebs cycle, we produce 2 as well. So yeah, if you sum up the ATP produced along the way with all these enzyme cofactors, you're gonna get more or less down to this number, 32 ATP. Some places you might find saying that is actually 38 ATPs. Other places might say that is a little bit less, because you know it's not something really precise. It really depends on a bunch of things. But for IMAT, 32 is your number. It's the thing you need to remember. All right, guys. For today, I'm gonna have only one question, but I realized that YouTube allowed me to put some questions to test you all. So I'll be trying this new features with other questions of oxidative phosphorylation, so you can try to test your knowledge into it. 2023, we had this question saying, which of the statements or set of statements about oxidative phosphorylation is correct? Number 1 is saying that complex 1 of the electron transport chain transfers electrons from FADH2 to coenzyme Q, because you guys know that complex 1 do not receive any electrons from this fella here, but from NADH. Statement 2 saying that ATP synthetase utilizes the energy of the proton gradient to catalyze the conversion of ADP to ATP. That's basically it. That's basically the chemiosmosis. Number 3 is saying that oxygen serves as an electron acceptor in complex 3. Almost. Complex 4 is the one responsible for that. And number 4 is saying that complex 4 receives electrons from NADH, which is wrong. Just remember, complex 1 and complex 2 is the only one that receive electrons from those cofactors. The other ones are just passing on. So, alright guys, that was pretty much it. I hope you guys enjoyed the video. I hope it made myself clear. If you have any questions, don't hesitate, just shoot it down in the comments and I'll be glad to help you. Don't forget to like the video and subscribe to the channel if you're new, and I'll be seeing you all in the next one.